In Sheep Brain Dissection Lab 3, we will make horizontal sections of the right half of the brain at 3 different levels and identify and draw structures visible at each level. Horizontal cuts are produced by placing a knife at the anterior end of the brain and slicing back to the posterior end. We will start from the top of the brain and cut sections toward the bottom. So those are examples of horizontal sections. So some of the sections you can see that they are differently oriented for convenience of showing structures. With first cut, we cut a large chunk of the cortex, and we now are exposing the level of the hippocampus. So this part here is the actually hippocampus, and hippocampus is here in the lateral ventricle in posterior limb. All surrounded here is cerebral hemispheres with the dorsal part and the ventral part here. So this second cut is horizontal section through the basal ganglia. And so basal ganglia is here, and you can see the striations here very well. That's why it's called corpus striatum. So this is internal capsule, and so this part will be thalamus. So those are cerebral hemispheres here. So third level is at the level of the midbrain, and also you can see some other structure we identified in previous sections. So this section have different orientation than previous one, but in this section we can see very well lateral ventricles. So this is anterior limb and this is posterior limb, and actually this structure here is hippocampus. So this structure here is nucleus caudatus. Then we have internal capsule, and this part here is the midbrain, where we have superior and inferior colliculus. This concludes Sheep Brain Dissection Lab 3.